welcome you all in this new Cryo-EM course. Last class we discuss about how we can get an image. We use the ray diagram, we learn how to draw the ray diagram, what is the role of the aperture, and now also that focus, different kind of focus and defocus. So now we are going to study little bit more about that focus, because that is one of the most important part. In the next class we will discuss little bit more about how to control control the dose rate, the biological sense sample. So today I will not go to that part. Today we will just discuss about little bit more about how to do the focusing. So you all know that focus means objective lens, right? Objective lens is connected with your focus. So that we already discussed, how objective lens we can use it for focus purposes. Now we are constantly talking about the converge and diverging electron beam, and also that converging and diverging electron beam, it is depending on the field strength, how much strength do you have in that particular electromagnetic lens. So whenever we are talking about that, okay, there is an electron beam and electron beam can come and converge in one particular point, we can always tell that, okay, so this is the converging or crossover point, and this is giving us an image. So now the same magnetic field or magnetic lens we are using, and then I am changing the crossover point. Crossover point is coming somewhere here, and if this is my image plane, it will continue in this direction, right? Now one more possibility is that I can use much more stronger magnetic field. If there is a much more stronger magnetic field, means it is pushing that incoming electron beam and it is convert. So you can see that crossover point is gradually going up. So crossover point can go gradually up and down. So when it is gradually going up and the electron rays, it will come and go like that, it will give us an image in this image plane. So one more possibility, if we are using a very less magnetic field. So let us erase this one, very less magnetic field. So there is not too much pressure. So this is our one of our electron beam. It is one of our electron beam. It is coming and merging somewhere here, right? Now we are talking about that we have a very low magnetic field. If we have a very low magnetic field, what kind of image do we get? So you can see images are going like that. If we have a almost no magnetic field, you can see electron can go like that. If you have a more lesser electron beam, you can see electron can come as almost parallel. So that was I was talking about when we are discussing that, okay, we have a condenser lens too, and the upper objective lens, these two combination, sometime we can make the electron beam parallel. So it is based on the what kind of magnetic field we are using, and then we can try to make it parallel. So if we have a C1, our electron beam will converge in one particular point, right? So this is our spot size. This is our spot size that we are going to discuss in the next class. Then our intensity. So intensity, if we just regulate this one in such a way, so this one my C2 or intensity, right? So intensity, if we regulate this point in such a way that electron beam will come out, and then there is another magnetic field which is regulating the beam. So something we are using some kind of magnetic field, that beam will come like that. So even if it is a lower lower objective lens if this is a lower objective lens, we are, and if our sample is somewhere here, you can see this upper objective lens current and the C2 we are using in such a way that it will make that electron beam parallel. So it will go through our sample as a parallel electron beam. It is not going to affect scattered or unscattered electron. So now if you just consider that we are using a very strong magnetic field here, a very strong current. So what will happen? Your converging point, it will go up, or crossover point, it will go up. If crossover point, it will go up, the electron beam will come like this, and no matter what, your upper objective lens and C2 cannot make the beam parallel. Or in the other hand, if we are using a very loose magnetic field, something here, that is also going to affect your electron beam, and it is going to expose your sample completely. So that is the reason we have to use a particular magnetic field which is helping us to converge the beam in such a way that next lens can take over these things. That is true for the spot size also. That is true for the C2 lens also. So that is going to help us. So now I hope you are getting enough example that why I am telling that C1, C2, objective lens all are fixed lens, and we can only change the magnetic field to change the crossover point or convert how the beams are converging, and that is helping us to regulate the magnification or any other parameters. It may be beam intensity, it may be the focus, defocus, magnification. So it is a fixed lens, but a different kind of magnetic field is going to regulate this parameter. Now come to the focus. So we want to discuss about the focus. So whatever that rays is coming out from the sample, there will be a no change, it will be as it is. Let's take a look to the other part. There will be a 3 different kind of parameters that we have to pay attention during this. So in all the cases, one thing, first we will make it fixed, that we have a lens here. We have a lens, and let us make this thing final, that this is my imaging plane. So that imaging plane will be fixed for all, so that height or everything it will be same for all this image. What we can do, we can draw a little bit smaller size of that object. So here consider that rays are coming. So for all the sample, this will be the same, right? If we just consider this is the start point, for the 3 different condition is possible. So here let me draw these things in a little bit proper way. Okay. So now if we just consider this is our start point, here also this will be our second point, here it will form that image, here this will also form that 2 image, right? Now rays are coming from the sample. Rays are coming from the sample. Okay. Same thing is true for this one also. Rays are coming from the sample here. Okay. So, now as we mentioned, we have a magnetic lens, electromagnetic lens, which is going to regulate the magnetic field. We just consider this is lens 1, this is lens 2 and this is lens 3. So, in one particular case we consider that okay, this is our, we are using a magnetic field in such a way that if this is our optical axis, this is our optical axis, our all the rays after focusing, it is going and merging in this point. Same thing is true for here also, rays are coming and merging one particular point. This is called focused image. Focused image. Now, we are using a magnetic field in such a way that okay, all the rays can come here and merge in particular one position, that is called the focused image. One more possibility is this one, that I will just use this ray and we consider that magnetic field we are using in such a way that it is converging somewhere here, and same thing is true for this particular ray. Okay. So, you can see it in blue case, rays are coming and converging in one particular point, that is called the focused image. For the red, I am using a stronger magnetic field, and that is the reason this red crossover point is going up. So, here you can see in blue the crossover point is somewhere here, for the red the crossover point it is going up, and it is when there is a crossover point, but image it is giving here. This kind of images it is called, this type of images it is called over focused image. So, this is our over focused image. Now, what about the last possibility? Last possibility, I will just use a different color here and show you that we are using a magnetic field in such a way that very weak magnetic field, and it is going in this direction, cross the image plane and converge somewhere here. This is called under focused image. So, here I will just draw that optical axis. So, this is basically optical axis. Here also, if we just consider, this is our optical axis where the image formation is there. Okay. So, it will be a straight line where you can see how this image formation is. Now, if you look at in this particular position, in all the cases until here the beams are behaving in the same way, they have a same angle. Then using this different magnetic field, basically we are converging the electron beam at different position. And now if we just try to measure this one, this angle, that okay, this is a alpha 1, this is our lens system, that is I mentioned lens 1, lens 2, and this is basically our object, and this is basically our image. Our image is basically, we consider that here also this is our image, this is our object, this is our image. Now here if you just measure this alpha 1 and also if you just consider this is our alpha 2, there is a angle difference. This angle, if you just consider this angle, it will be not same as this. So here this alpha 2, it will be over focused image. If you just consider that over focused image, so alpha 2, it will be smaller than alpha 1. So this alpha 1, this angle, it is, this angle means this alpha angle here, this alpha, it will be much bigger than alpha 2. So how this beam is converging and how it is coming and merging, it is depending on this, and that is also regulating what kind of images we will get. This kind of image we will get the under focused image. So under focused image, this angle alpha 2, it will be smaller than alpha 1. So one more last things for today's class, let's take a look. So this is the focusing, and now what about if we just go for a, we are talking about the converge, diverge, how it is converging and diverging the electron beam. So we'll go to the PPT and discuss little bit about the different types of aberration, because that is one of the most important part of our electron microscope, and that is we have to know that whenever we are talking about the lens system, how lens is working, how lens is converging the electron beam or diverging the electron beam, it is directly connected with a different types of aberration. So initially we will start the different types of aberration, and then we will go for a ray diagram and try to understand how this aberration is playing an important role in our making the coherent electron beam or giving a different types of images. So what kind of aberrations are there? So let's take a look. So there are majorly there are 6 aberration we are getting it. There are many others aberrations are also there. We will discuss about those things, how we can classify those aberration, and those aberrations is basically going to affect our signal also most of the time. Signal means when we have some so we have a parallel electron beam. Parallel electron beams are coming and interacting with our specimen, right? This is our specimen. So all this parallel electron beam come fall to our specimen, and then they will deflect and give us an image. Now we are always considering everything is in an ideal situation when the parallel, whenever we are considering all the beams are parallel, we are considering everything is parallel. That is how we are drawing our ray diagrams. So all are parallel. Then there is a deflection. So when deflection is there, there is a scattering. So unscattered electron beam and scattered electron beam. So they are coming together and they are giving some kind of phase difference and giving us image. But most of the cases our lenses are not in an ideal condition. So there will be some kind of differences, and even if you consider the glass lenses, so glass lenses is not uniform, it is not symmetric. Same thing for that electron microscope also, and also it is a very difficult task to put so many electromagnetic lenses. If you just think about the engineering part, it is a very difficult task to place those electromagnetic lenses, one by one together. So it is not that an easy task, and then control all this electron beam properly. There will be some kind of aberration, and that is going to affect our images. So what kind of aberrations are there? Let's take a look. So aberrations, as I mentioned, there will be 5 major aberrations we are always considering. That is our spherical aberration, then chromatic aberration, astigmatism, that I use that term couple of times, that astigmatism, then coma aberration, distortion and curvature of the field. So let's take a look about those important part, how we can classify those aberrations. So if we just go to the next slide, you can see it that aberration we can split it into two halves. So one is geometrical aberration. So this slides or this diagram is taken from so there is a electron microscope made by Geol, and this slide is taken from the Geol website. So if you just go to that Geol website, you can see these different types of aberration. So in the schematic diagram you can see so there is a geometric aberration and the chromatic aberration. So if you look at the geometric aberration is split into two halves. So we are more interested about this 5 aberration, that is called the Siddle aberration. The Siddle aberration name came from that Phillips von Siddle. So now here we are more interested about this 5 aberration, where there is a spherical aberration is coming, then chromatic aberration, astigmatism, curvature of the field, and the distortion. And chromatic aberration also, it has a 2 different part. One is the axial chromatic aberration and magnification chromatic aberration. So we will not split all of these things in one half. Majorly we are going to discuss about that chromatic aberration, and then we are going to discuss about the spherical aberration, coma, astigmatism, and curvature of the field, and distortion also. So let us discuss about the chromatic aberration at the beginning, and then we will talk about spherical aberration. So what is that chromatic aberration? In glass lenses this is a very common problem. So if we have a glass lens, here, and if this is our optical axis, so we all know that we already discussed about that visible light, wavelength nanometer to 750 nanometer. So we discuss about all of this. So within this range, what rays are there? We have starting from blue, all these 7 different colors will be there. So here we are talking about blue, and then green and red. So based on the wavelength, if our lens is not perfect, it will split all these rays based on their wavelength. Okay, so what is going to happen if there is a blue rays? So blue rays, here I am just so let us draw the normal ray, and normal ray color is only black. So it's a black color rays are coming, and which is going to merge, but due to some kind of defect in the material of this lens, glass lens, it is going to split. So here when these rays are traveling inside this glass lens, so there is a different types of material, and so red light has a different wavelength, green light has a different wavelength, blue light has a different wavelength. So those rays will split here inside that lens, and when they will come out, so they will just merge in a different point. So blue rays will merge like that, then green rays, it is going to merge somewhere like that, and then red, it will merge further like this. So now you can see when this will give us an image here, it will be combination of all these 3 or 4 rays, and it is going to reflect here in your image, where you can see sometime blue or red boundaries outside, or if you are just trying to take someone's face, so sometime it is too much reddish, or sometime it is bluish. So because of this chromatic aberration and this not perfect lens we are using, materials is difference of this lens, it is splitting all these rays in a different form. It is splitting blue, green, red, and everything is going to reflect in your image. Similar problem is possible when we are doing the electron microscope. Here what is the major parameter? Major parameter is the wavelength. So lens is deflecting the wavelength So, blue has a different wavelength, green has a different wavelength, red has a different wavelength. All are coming and merging in one particular point and giving us a different color images. So, in electron microscope we are considering that okay, all the electrons are coming into particular this point and all the electron has the same energy, that is called E. Now, if some electron has some extra energy delta E, or if some electron has this kind of delta E, either plus or minus, then what will happen? We are considering all these electrons are coming and merging in one particular point, because this E is fixed, the energy of incoming electron is fixed. If, because here we are using a fixed magnetic field, if the energy of the incoming electron is not fixed and we have a fixed magnetic field here, then electron can merge in a different plane. So, it will not go all the way this, it will merge somewhere here or it will merge somewhere here. So, energy is directly connected with your wavelength and that is going to reflect when you will do the imaging using electron microscope. So, today's class I will stop it here. We will discuss these things in next class in details, all the aberration including this chromatic aberration also. Thank you all for your attention.